I'm going to be talking today about some of the work I did during my PhD, where I was looking at the link between the DNA damage response in E. coli and CRISPR-Cas adaptation. So CRISPR-Cas is an adaptive prokaryotic immune system that's used as a defense against invasion from bacteriophage and mobile genetic elements, so very abundant, found in about 2/3 of the prokaryotic genomes. So the system is split into two different arms. So the interference arm, which I'm sure you all have heard of, the Cas9 protein, everyone's favorite a few years ago. So this is involved in interference, which is involved in the destruction of the invading elements. Whereas what I and our lab work on is adaptation, which is the process by which immunological memory is generated. So I've got a little diagram here that shows a CRISPR array. So there's a leader sequence here with the white box labeled L, and this contains elements that guide the site of integration. So the CRISPR array is composed of a series of repeats and spacers. So these spacers are small fragments of DNA from the invading element that are used by the interference machinery as a template to destroy complementary DNA upon future infection. So for E. coli, adaptation is catalyzed by the Cas1 and the Cas2 protein. They form a complex together. Cas1 does the catalytic activity, catalyzes the integration event, whereas Cas2 acts as a molecular ruler that controls the length of spacers to ensure that every spacer in the array is the same size. So to start with, I wanted to look at the effect that Cas1 and Cas2 expression has on the cell. So this first image here is just a wild type E. coli under a light microscope, and you can see it's a classic rod shape. So then in this next image, I overexpress Cas1, Cas2, and you can see that the cell morphology changes a lot. Massive cellular filamentation is induced. So I also did some control experiments where I overexpressed either Cas1, either Cas2, or a catalytically inactive dead Cas1, Cas2. And you can see all three of these controls didn't change the cell morphology. So this shows us that the catalytic activity of the Cas1, Cas2 complex is affecting the cell in some way. So we thought the most likely reason this would be due to DNA damage by the catalytic activity. So the DNA response in E. coli, the SOS response, is the master regulator is the LEX-A repressor. So this binds to the promoters of genes of the SOS response and acts as a physical barrier to prevent RNA polymerase from expressing the genes. Upon sensing DNA damage, REC-A is activated, forms filaments on the damaged DNA, which then mediates proteolysis of LEX-A. The cellular concentration of active LEX-A then decreases, resulting in a de-repression of genes of the SOS response and then switching on of the SOS response. So one of the genes we're most interested in the SOS response is SOS. So this inhibits the polymerization of FTSZ, so higher order structures aren't formed, so the Z ring doesn't form, so the cell filaments and gets longer. So I thought the most likely explanation as to why Cas1, Cas2 is causing filamentation is because it's inducing SOS expression. So to study this, I used a SOS reporter plasmid. So this contains a GFP gene downstream of the SOS promoter. So that means that when native SOS is expressed from the chromosome, the GFP will also be expressed from the reporter plasmid, and you'll then be able to use the GFP readout as a proxy for SOS gene expression. So I then induced Cas1-2 expression across a range of arabinose concentration, and you can see the higher the level of Cas1-2 induction, the higher the consequent level of SOS expression. So therefore, Cas1, Cas2 is causing cellular filamentation by activation of SOS. So I wanted to study what genes of the SOS response are the key genes involved in CRISPR adaptation. So to do this, we used a CRISPR adaptation reporter strain we made in our lab. So there's a constitutive promoter upstream of a reporter array, which contains a stop codon. This stop codon prevents the expression of our reporter gene, LAC-Z. So when we grow on, do a blue-white screen, those cells with a stop codon obviously produce a whitish yellow colony. So when adaptation happens, an extra spacer repeat unit is added to the array. Now this is 61 base pairs, and that causes a frame shift, which results in the stop codon being moved out of frame, our reporter LAC-Z gene being expressed, and so the cells then can metabolize lactose and become blue. And so here's a picture of a papillation plate, this assay. So these are left in the incubator for 5 days, and you can take photos of the colonies, and each blue spot, which is called a papillae, marks a CRISPR adaptation event. So this allows different conditions to be compared, as the number of papillae per colony is a quantitative measure of spacer acquisition. So as I said earlier, the LAC-Z gene repressor is the master regulator of the SOS response. There's a mutant, LAC-Z3, which contains a substitution in the cleavage site. So regardless of how much DNA damage there is in the cell, the SOS response can't be induced. So I transduced this allele into our reporter strain, and as you can see, the number of papillae, the number of blue dots on the LAC-Z3 mutant is lower than on the wild-type cells. So by removing the ability to induce the SOS response, the rate of CRISPR adaptation goes down. So another way to inhibit the SOS response is to inhibit the activity of REC-A. So you can do this genetically. So REC-X is an antagonist of REC-A function and prevents the formation of REC-A filaments, which then prevents the consequent induction of the SOS response. So I clone the REC-X gene into an inducible expression vector, and as REC-X expression increases, you see the number of papillae, the blue dots, decreases, and consequently the number of adaptation events decreases. So this is a genetic way to inhibit REC-A activity. You can also do this chemically. So zinc inhibits REC-A ATPase activity, which is required for LAC-Z cleavage to induce the SOS response. So I performed assays across a gradient of zinc chloride, and the same again, increased zinc, increased inhibition of REC-A, the rate of CRISPR adaptation goes down. So I'm aware that's a lot of gene names I've just talked at you. So just a little summary. So Cas1, Cas2 causes cellular filamentation by inducing the SOS response. If I use antagonists of the SOS response, the rate of CRISPR adaptation goes down. So I next wanted to see what happens when I induce the SOS response. So if I grow in the presence of antibiotics, the literature tells me that these 4 antibiotics, so ampicillin and cephalexin, they're both beta-lactam antibiotics. Also nalidixic acid and mitomycin C, these all induce the SOS response. So these, the first row here is with no antibiotic, and then as we move across, the concentration increases. And you can see that for all 4 antibiotics, the number of papillae per colony, the number of blue dots increases. So if we're inducing the SOS response, we're getting more CRISPR adaptation. But I then wanted to just check that in this system, the concentration I'm using is actually inducing the SOS response. And interestingly, despite what the published literature says, I found that in my system, the beta-lactam antibiotics don't induce the SOS response. So these must be increasing CRISPR adaptation by an alternative mechanism. Whereas the ones that target DNA, nalidixic acid, and mitomycin C, these do induce the SOS response. So I think it's likely that the mechanism by which these increase CRISPR adaptation is through inducing the SOS response. So if you remember from earlier, so the LAC-Z3 allele prevents induction of the SOS response. So I wanted to look at the effect that the antibiotics have in our reporter strain when this allele is present. So the ampicillin and cephalexin, these don't really appear to have any effect on the rate of CRISPR adaptation. There's a slight decrease at high concentrations. However, the DNA targeting antibiotics have an immediate effect and the rate of CRISPR adaptation goes down, which is similar to the slide we saw previously that suggests that these two antibiotics increase the rate of adaptation because they induce the SOS response. So I've shown that if you inhibit the SOS response, CRISPR adaptation goes down. If I activate it, CRISPR adaptation goes up. So I wanted to determine what the key gene of the SOS regulon is. So there's approximately 60 genes. So I went through and I spent some time looking at what the different function of the different genes, and I thought the most likely gene to be causing our effect is the SOLA gene. Now I'm sure you remember from earlier, SOLA it inhibits FTSZ polymerization, which induces cellular filamentation. So I wanted to see what happens if I overexpress this gene in the absence of all other genes of the SOS response. So I cloned it into a inducible vector and performed population assays, and the rate of CRISPR adaptation increased, but it was most apparent on day 3 of day 5. So this shows that when I'm overexpressing SOLA, the CRISPR adaptation events are arising earlier in the colony's life cycle. So I then wanted to see what happens when I overexpress SOLA in the LEXA3 background. So, as I mentioned earlier, LEXA3 does not induce the SOS response, it's uncleavable, so there's no native SOLA expression. So I used the inducible expression vector to overexpress SOLA, and you can see that the rate of CRISPR adaptation increases along with the level of SOLA expression. So SOLA in the LEXA3 background complements the defect in CRISPR adaptation. So this shows that SOLA is the key protein in the SOS response to help support CRISPR adaptation. So that's the end of my talk, so I'll just summarize. Cas1-2 expression induces filamentation by activating SOLA as part of the SOS response. If I use antagonists of the SOS response, the rate of spacer acquisition goes down. If I induce the SOS response with antibiotics, the rate of adaptation goes up. And this antibiotic-mediated increase is not seen in a SOS uninducible background. So I'll summarize this years of my life in one sentence. The SOS response supports CRISPR adaptation due to the activity of SOLA mediating cell division and causing filamentation. So I just want to say thanks. So Ronnie, unfortunately, couldn't make it today, our supervisor. Jack, the former postdoc in our lab. Chris, who's here today. And then Wellcome Trust for funding this research. Cheers.